Ever wondered how scientists analyze millions of cells to fight diseases? Manually identifying each cell is slow, tedious, and nearly impossible on a large scale. But with computer vision, we can train AI models to do it in seconds. This is cell segmentation, automatically outlining every cell to measure its size, shape, and even track its behavior.
So now let's see how you can perform cell segmentation using YOLO. So what is cell segmentation mean? So let's take an example. This is the video I am taking as reference. This is a microscopic video of a tree plant cell. So if in biology you need to study the number of cells in particular frame or the size of cells, this kind of model will help us in that. So now let's see how you can do that. First of all, you have to take this video and convert into a black and white video, because black and white videos are much easier for computer vision model training. Now, then this is the notebook I have created for this particular for model training. You have to clone this repo which I have created. This repo helps us you to extract some random frames from this video for data annotation. I have run this and collected around five five frames. Now, these frames are randomly taken from the video, so we just have to annotate it and use that annotation in model training. Let's see how you can annotate it. Let's go to labeler. This is the labeler and annotation of my project screen window. You just have to select create a class cell and then use the uh, SAM annotation to perform segmentation. As you can see, it will segment this part now and use that for all also. So now your whole frame and of cells are segmented. Now you just have to accept it and export it. After export, you will get your annotation in this format. Here is the annotation for each bounding boxes. So now let's get to our model training part. First of all, you have to convert your annotation, which is this annotation JSON, using Coco to YOLO converter into your YOLO format. So what is YOLO format? Uh, this is the YOLO format. You have one directory for images, one directory for labels, and another directory called data.yml for parts to that data set, part to train value, part to validation set, and part to test set, and number of classes and name of that class. So this code will help you do that. Now let's get to our main functions of model training. First of all, you have to use torch to clear your CUDA if you are using GPU, as model training require VRAM, so your RAM should be, VRAM should be free. Let's run this. Now check the VRAM. As you can see, I have 0 allocated GB and 0 cache data. My 7 GB VRAM is free. Now let's see how you can train. You have to import YOLO from Ultralytics. This YOLO model helps you for model training using YOLO 11 M variant. Then you have to use model and its method train. Provide this data.eml file here and then train that model for 500 epochs on image size 640 and batch -1. This helps to select the batch size on its own, and then the device equals to 0, which is for GPU and the workers, and the name of that run is called model M. I have run this, and this will create a folder called model M, where there is your train model. It will take around 1 to 2 hours for model training based on your GPU. These are the two trained model I have gotten. Now let's see the class you have to create for inference and finding the area of that cell segmentation. Now you just have to initialize your model, confidence threshold and IOU threshold. So now you have to for a random color you have to just use NP random value for color creation. Now let's see what you need. You need a polygon center which helps to find the center of polygon, and there is the function to find the area of polygon, which is the segmented area of the cell. Now for process frame, which is our most important frame, you just have to use model and track, then give it frame by frame of the video and persist equal true, then there is your threshold and there is your IOU value which you got using initialization. Now for each results mask you have to just store that mask value boxes and use a color to print that marks over a frame. Here's a fill polygon and added weight function of computer vision libraries, OpenCV library. Now this is a simple part. You just have to use each result value and perform annotation or overlay over each frame. Here you can also put a rectangle dash to show the size of that uh, segmentation. Now and then there is a process video. This process video will run this process frame function on each frame one by one. Here is your now, and at the end it will save it. Now let's create a variable which used to store your model part. This is the model part. I'm storing it here. Now after that you have to just run an example uses. I am going with the model part, confidence threshold about 50% and IOU threshold about 30%. And here is a sample video. Then let's run this and see in live. Sorry. I let's run this. Now the referencing happening right now. As you can see, each frame is showing the cell boundary and the area, which is the pixel. You can choose your own unit based on your requirement and the microscopic value of that frame. Here is it saying this segmented area value 34,000 or 35,000 pixels squared. As you can see, it makes it much more easier to find the area of that cell. You can also add a function to find the length of total number of segmented value in each frame and show it here to find the number of cells in a particular frame. With this, you can perform cell segmentation.
If you'd like to explore practical resources and hands-on examples, we've created a dedicated GitHub repository. It's filled with cookbooks covering computer vision, AI agents, and more. Link is in description.